You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is January 15th, 2024. Our topic today is Abbas Chapter 5, Antibodies and Antigen, presented by Dr. Nikita Raje. She is an Associate Professor of Internal Medicine Pediatrics and is the Chief in the Section of Allergy Immunology at Children's Mercy Hospital.
Topic for today, Chapter 5 from Abbas. I'm Nikita Raje from Children's Mercy, Kansas City, University of Missouri, Kansas City. Here's my email if you have questions, and today is January 15, 2024. Let's get started. Here are my disclosures, nothing relevant to the talk today. Learning objectives for today: to expand the understanding of antibody structure, synthesis, assembly, and expression of immunoglobulins, antibody binding to the antigens, and then structural and functional relationships in antibody molecules. Couple of pre-test questions here. We'll come back to these. Which one of the following is a correct description of the basic symmetric core structure of an IgG antibody? And antigen binding site of an IgG antibody molecule is composed of which of the following? All right, so looking at the antigen binding molecules, we know that for the adaptive immune system, there are two antigen binding molecules. One is T cell receptor and another is immunoglobulin. Both of them are made up of three complementarity determining regions, and we'll talk about those. But in general, there are two heavy chains and two light chains in the immunoglobulin molecule, whereas the TCR has two chains. Both of them have the variable regions that make the antigen binding site. We know that the TCR can bind to a peptide that is presented to it on MHC molecule. However, the immunoglobulin receptor or immunoglobulin can bind to a variety of different macromolecules, which could be proteins, lipids, polysaccharides, small chemicals. So a variety of different molecules that they can bind to. They can also bind to linear or conformational determinants, whereas for the TCR, it has to be a small linear peptide. That's about it. If it has a conformational shape, it may not be able to bind to that. Or it may bind to just a part of that has linearity in it. The affinity can also vary quite a bit. And the immunoglobulins have a rapid on rate and variable off rate versus the TCR, which means the antigen binds to it quickly, and then some of them can come off pretty quickly. Some of them will take time. TCR, on the other hand, it takes it takes time for it to bind to the antigen, and then it takes a while before it comes off. For the antibodies, we know there are two types of antibodies. One are membrane bound, which could be on B cells. So the antibodies make B cell receptors, and those are immunoglobulin that are bound to the membrane of the B cells. Versus the antibodies that we typically talk about are the secreted ones that are not bound to a cell. There are 5 different classes of antibodies that are known: IgG, A, E, M, and D. About 2 to 3 grams of antibodies are produced every day, and 2/3 of which are immunoglobulin A, that is produced, most of which is produced by the plasma cells in the gut and released into the lumen of the gut. So the most prominent antibody, if you take the whole body, not just the blood circulation, is IgA. The functions of these antibodies include just neutralization of microbes or toxins. So they attach to the microbes and toxins so that they do not bind to anything else. The immunoglobulins or antibodies can also activate the complement system. That's what we know as the classical pathway of complement activation. Antibodies also help in opsonization of pathogens for phagocytosis. So once they are kind of tagged for phagocytosis, they are targeted by neutrophils or macrophages so that they can engulf them and then kill them once they are in the phagolysosome. Remember, we talked about the antibody dependent cytotoxicity, which is ADCC, that is performed by the help of antibodies by NK cells. This is what we talked about in last chapter. Also, the mast cells, once they have IgE antibody bind to its receptor on the mast cells undergo degranulation, which is the function of the mast cells. So these are the different types of functions that antibodies perform. The important part of understanding the function of antibody is to understand the structure of these antibodies. So the antibodies are made up of two identical light chains and heavy chains that have something called immunoglobulin domain. We'll come back to talk about what the immunoglobulin domain is made up of. But in general, these two identical light chains and two identical heavy chains together form two parts, variable region and constant region. The variable V region is also known as antigen binding region, and the reason it's called variable region is the structure of it is pretty variable, but that variability is important for recognition and specificity, as well as the diversity of antigens that it can bind to. Versus the non-antigen binding region is also known as the constant region or C region. It's not so variable, but it's important for the effector function. And it's also responsible for the physiochemical properties that these antibodies have. So this constant region is the one that determines the class of antibody, which is IgG, A, E, M, right? So each of them have particular type of physiochemical properties, and they have particular type of effector functions, and that's because of the constant region or also known as the Fc region or fragment C of the antibody molecule. So let's look at the structure more in depth. So the antibodies have a structure called immunoglobulin domain in them. Each chain has series of these repeating domains, immunoglobulin domains, which are made up of 110 amino acids. Each of that immunoglobulin domain, it has two layers of beta-pleated sheets, which run. There are 3 to 5 strands of antiparallel polypeptide chains. So they run in antiparallel direction, and these layers are bound to each other by disulfide bridges. The adjacent strands are bound to each other by short loops. And these short loops are also known as CDRs or complementarity determining regions. These are important because this is where the amino acids that make up these chains are most variable. So these CDR regions are most variable in terms of the antibody molecules. Now this structure overall, where there are 110 amino acids making up the immunoglobulin domain. This immunoglobulin domain has 2 layers of beta-pleated sheets, and each sheet has 3 to 5 strands of antiparallel polypeptide chains. The overall structure then can fold onto each other to give it a shape, a different shape that is seen in here. Let's see here. And that globular structure is because of the folding of these 2 beta-pleated sheets to give that Ig domain structure to it. Sorry. So this particular structure is not only present in the immunoglobulins that's seen here in the figure, IgG molecule, but it's also present in other molecules or receptors, and all of them together are part of what is known as immunoglobulin superfamily, which means that immunoglobulins are part of the immunoglobulin family, but there are other molecules that also include chains that give this Ig domain structure to them, such as in T cell receptor, MHC molecules, co-receptors like CD4, ICAMs, CD28, all of these have this Ig domain structure in it. And so they are all part of the immunoglobulin superfamily. So important structure to remember. Now looking at the antibodies, we know that there are 2 light chains and 2 heavy chains here in red, the light chains in green, and the light chains have 1 variable region and 1 constant region. The heavy chains have 1 variable region and they have 3 to 4 constant regions. The antigen binding site is formed by the variable chain or variable region of the light chain and the variable region of the heavy chain. So there are, in one molecule, there are 2 antigen binding sites, whereas the effector function comes from the heavy chains, and typically it's the heavy chains, the CH chains, that also determine what type or class it is. These heavy chain constant regions are bound to each other, and there is a region in between CH1 and CH2, which is called the hinge. Apart from that, there are covalent and non-covalent bonds, interactions between these chains so that they are kept next to each other. So the light chain, the variable region of the light and heavy chain and the CH1 region of the heavy chain and the constant region of the light chain, which is just 1, right? All of that together forms the antigen binding fragment. It's called the FAB region. And then the rest of the constant regions form this FC region of the antibody structure. Now, if it's a secreted form of the immunoglobulin, it has a small tailpiece at the end of it. However, if it's a membrane-bound antibody, such as IgM that is on the B cells, it needs a longer tailpiece that can be inserted into the plasma membrane of the B cells. All right, so how was this structure determined? So they use the rabbit immunoglobulin and use some enzymes to break down the structure of the immunoglobulin. When the enzyme papain was used, the immunoglobulin structure or the molecule was broken down so that it broke down above the hinge. So it broke down into 2 pieces that had each having 1 antigen binding site, one FAB molecule. Similarly, the second FAB, and then the third fragment was the FC region. However, if pepsin was used, the breakdown was below the hinge, because of which it gave rise to 1 big fragment that had both antigen binding sites and smaller peptide fragments that were released from the FC region. So an easy way to remember is papain that has 3 vowels gives rise to 3 pieces, big pieces, versus pepsin, which has 2 vowels, gives rise to 2 parts. One is the whole FAB region with 2 antigen binding sites, and then the peptide fragments. So again, something to memorize here. Remember, anything that's in red is the part that I want you to memorize. Let's talk more about the antibody variable regions. So we talked about how that beta pleated sheets have antiparallel strands that are connected to each other with short loops. And those short loops have the most variable amino acids. So those are called CDRs, or hypervariable regions. And those hypervariable regions are numbered, so CDR1, CDR2, CDR3, and so on and so forth. The variability, if you look at that variability, you see that the variability is much more in these CDR1, CDR2, CDR3 regions rather than the rest of the antigen binding sites on the antibodies. So Let's look at that. So it is confined to those regions, but that includes both the variable region of the light chain and the variable region of the heavy chain. Each region is complementary to the antigen shape, and that's the reason it's called complementarity determining region. Each of these CDRs is 10 amino acids long. And the most variable CDR out of this is CDR3. All right, we talked about the variable regions. Now we talked about the constant region. As we talked about, these constant regions are the ones that determine the class or the isotype of the antibodies. G, A, M, E, D. G and A also have subclasses. So IgA1, IgA2, IgG1, 2, 3, and 4. The constant region is given the name. So if it's a IgG antibodies, it has gamma chains. If it's A, alpha chains. Typically G and A have 3 CH regions versus M and E have 4 mu or epsilon chains. These determine the proteins that they interact with the Fc receptors. So Fc receptors are present on various cells. They may be present on NK cell, they may be present on macrophages, neutrophils, and depending on what Fc receptor it has, it can bind to, or mast cells, for example, it will determine what immunoglobulin it can bind to. So say IgG is opsonizing a virus or a bacteria. That IgG can interact with a macrophage by binding the Fc region of the antibody to the Fc receptor gamma on the macrophage. Or if it's a virus, that IgG using its Fc region can bind to the NK cell, so to the CD16 on the NK cell, so that it can give rise to antibody-dependent cytotoxicity. It could be an IgE that binds to Fc receptor epsilon on mast cell, and because it's bound to some antigen or say a parasite, it can bind to the Fc receptor on the mast cell and cause degranulation. So these are just the examples of how the Fc region of the antibody determines what cell it can interact with, and because of that will determine what type of effector function it is, right? Because if it's a mast cell and it's bound to Fc epsilon receptor, it can cause degranulation and histamine release, versus if it's NK cell and if it's a virus, it can cause antibody-dependent cytotoxicity. So all those effector functions are dependent on this constant region. So hope that makes sense why these classes are important, and how the constant region will determine what kind of cell it interacts with and what kind of effector function we get from the antibody. These are, again, just tables showing summarizing what we already talked about. Few more details: IgA, IgD, IgE. If you look at the half-life, it's pretty short, few days, 2 days to 6 days here, varying between different classes of antibodies. Something to note for IgA is that the most common form you will find these antibodies in are a dimer. They can also be monomers or trimers, but most of the times the two molecules of IgA are bound to each other by something called J chain. We know the function of IgA is mucosal immunity. IgD is present in B-cell antigen receptor. It plays a role in inflammation as well. IgE is defense against helminths parasites and can cause allergic reactions. And then IgG, it is present as a monomer. There are subtypes of IgG, subclasses G1 to G4, and of course we know different types of functions that IgG perform, including opsonization, complement activation, ADCC, neonatal immunity, and then it also provides feedback inhibition for B cells. IgM can be present as a pentamer. So again, a J chain that keeps 5 molecules of IgM together. It's the receptor for B cells, and it also helps with complement activation. So something to note here, IgG antibody has the longest half-life, about 3 to 4 weeks. All right, the hinge region. So why is there a hinge region in the structure of the antibodies? It's present between CH1 and CH2 heavy chain, right? So here is the light chain that has the variable region and the heavy region, variable region and the constant region of the variable light chain. And then the heavy chain has the variable region that binds to the antigen, but the constant region has CH1, CH2, CH3, in some cases CH4. Between CH1 and CH2 is where the hinge region is. It's 10 to 60 amino acids, maybe even more than 60 amino acids in length, and the function of this hinge region is to provide flexibility, which means, say there is an antigen that has repeating epitope. It can bind to those epitopes using these two antigen binding sites on the same molecule, but depending on how close or far away these epitopes are, the hinge region can give the flexibility so that it can bind to them. We talked about the constant regions and the heavy chain regions that determine the classes of antibodies. There are also light chain classes called kappa and lambda. In human antibodies, 60% of the antibodies have kappa light chains versus 40% have lambda light chains. That ratio is really important because it changes in malignancies, B-cell malignancies. We talked about the monomeric versus multimeric, so dimeric or pentamers that are formed in IgA and IgM. Sometimes IgM can have hexamers as well. Why is it important to know? Because multimeric, so either dimer or pentamer or hexamers or trimers for IgA, all of these can increase the number of antigen binding sites, and that can, even though the affinity of each site may be the same for monomers versus multimers, the avidity with which that antibody can bind to that antigen can be increased with the help of dimers, trimers, pentamers, or hexamers. We talked about how these dimers or pentamers, the molecules are kept together by something called J chain, that stands for joining chain. All right, we talked about this tailpiece part of the antibodies that determines whether it's bound to the membrane or it's secreted. So in cases of IgM and IgD that make the B cell receptor, the antibody is bound to the B cell with the tailpiece that has a smaller tailpiece than the IgG that's bound to the cell, but it's longer than the secreted form of IgG. So secreted form of Ig has very small tailpiece, versus the IgM, IgD have a longer tailpiece. Compared to that, the membrane IgG has a longer cytoplasmic tail. All right. Talking about the different types of antibodies or differences in the antibodies, if there is a transfer of, well, before I say that, so in the blood, there are circulating antibodies, and if the blood clots, the fluid that's left behind, which is the serum, is where all the antibodies will stay, and that doesn't have the coagulation factors. Those coagulation factors are in the clotted part of the blood, but the antibodies stay in the fluid part, that's the serum. That serum, if the serum is serum antibodies are introduced from one species to another, they are seen as foreign, and so there is an immune response against those foreign antibodies. So the antibodies that are formed are typically against the C region of the antibodies. And so if there are serum antibodies introduced from one species to another, it can lead to illness called serum sickness. There are differences in the antibodies that are present in antibodies from one individual of a species compared to this another individual of the same species, and the antibodies that would be produced against those antibodies are called anti-allotypic antibodies. There are also differences between antibody variable regions that constitute idiotypes of antibodies within a same person. They are called anti-idiotypic antibodies. Another important concept to understand is monoclonal antibodies. So antibodies of this identical structure and the same specificity are called monoclonal antibodies. How are these produced? So we knew that the antibodies coming from, say, a myeloma would be monoclonal because they have the same identical specificity. But those could be against various specificities, and it's difficult to determine which type it is. However, this process was used by scientists, Kohler and Milstein, to form something called hybridomas, so that you can produce monoclonal antibodies of desired specificity. So how does this process work? So say there is a mouse model where you introduce an antigen X. That mouse is going to produce antibodies, so B cells that lead to production of antibodies against that antigen X. If you want monoclonal antibodies against that antigen X, you can use those B cells and fuse them with myeloma cell line. That will give rise to these hybrid cells that have qualities of the myeloma cells, so that they are immortal, and then they have the quality of the B cells that are specific for producing antibodies against antigen X. So these fused cells or hybridomas then give rise to monoclonal antibodies against that antigen X. This type of monoclonal antibodies are used in clinical applications, in immunodiagnostics, in identification of tumor antigens, in therapies. So monoclonal antibodies that are used to target different antigens or cells, and then also functional analysis of cells and secreted molecules. Based on these monoclonal antibodies, you can recognize various cell populations. And so now the cells that are recognized by a particular monoclonal antibody can be used to define the cell, and they are called clusters of differentiation, right? So we use CD markers based on the monoclonal antibodies that can be used to recognize different types of cells. So to make sure that we follow a particular way of naming monoclonal antibodies, there is a nomenclature that has been adopted, right? So we'll talk about that. This basically talks about how there is a limitation of monoclonal antibodies, because typically when you have the mouse hybridomas that produce antibodies, monoclonal antibodies, there can be development of human anti-mouse antibodies that can be a problem. Genetic engineering can also be used to obtain cDNA from the hybridomas and from the myeloma protein to give rise to hybrid genes. So based on what part of the monoclonal antibodies is from human and what part comes from mice, there are different names or suffixes to the monoclonal antibodies so that you know which type of antibody you are talking about. So if it's a murine antibody, so completely coming from mouse. Does anyone know what's the suffix used?
OMAB?
Yep, OMAB. So examples muromonab or blinatumomab. So OMAB is the suffix that you would look at and say, okay, that's a mouse antibody. Chimeric, which means the variable regions are murine, but the rest of it is human. Anyone know what the suffix is?
ZMAB.
ZMAB, infliximab, for example, right? Rituximab. So those are the examples of chimeric antibodies. Humanized antibodies, which means even most of the variable region is human, but the CDR region is from mouse. What's the suffix?
Zumab.
Zumab. So omalizumab, pelivimizumab, so tocilizumab. So a lot of these antibodies that are humanized, which means only the CDR regions are from mouse. Benralizumab, right? So those are all humanized. And then human antibodies. What's the suffix?
Umab.
So umab, so something like adalimumab. So all of these are different types of suffixes that you have to think about to determine what part of the monoclonal antibody is coming from mouse and the rest from human. So these are the examples again of various monoclonal antibodies. Not going to go into the details, but something for you to look at. All right, how are these immunoglobulins synthesized in the human body? So the heavy chains and the light chains, like other proteins, are produced in the ribosomes, right? On the rough ribosomes that are present on the endoplasmic reticulum. Once those heavy chains and light chains are synthesized, those chains have to be folded with the help of chaperones, and that's done by the endoplasmic reticulum. Once that happens, they are packed in vesicles and they are modified in Golgi apparatus. So they are modified so that they get, more structure added to it, so that so carbohydrates that are added, and then that Golgi apparatus can then help to release those immunoglobulins, take them to the plasma membrane. And if these are membrane-bound immunoglobulins, they are going to stay attached to the plasma membrane, versus if they are secretory, they are going to be transported out. So based on the antibodies that are bound to the plasma membrane, for the B cells, the B cell maturation can be determined at what stage is that B cell in its differentiation. So the B cells mature and go from being a stem cell to being a fully activated or mature B cell, and then a plasma cell. And based on the presence of surface markers, you can determine the stage of maturation. So the stem cell, of course, will not have any immunoglobulin production, but when it becomes a pre-B cell, a stem cell that is determined to be a B cell will become a pre-B cell, and at that point, there will be cytoplasmic mu heavy chain in the cytoplasm of that cell. There will also be pre-B receptor, so that receptor is going to go to the surface. When the mu heavy chain goes to the surface and then gets expressed with surrogate light chains, that's still pre-B cell. For it to become an immature B cell, the signaling through the pre-B cell has to go through so that it can survive longer, and then the true IgM antibody receptor will be present on the surface. When that happens, that cell is called immature B cell. Along with IgM, when there is IgD expressed on the membrane of B cells, they are mature B cells. Can you hear me? Mm-hmm. Okay. And when that mature B cell comes in contact with the antigen that it can bind to, it's called activated B cell. Once it's activated, it will differentiate into plasma cell, which has an eccentric nucleus. It changes shape, and then it can release antibodies that have the same specificity as the IgM present on the membrane of the mature B cell. So stem cell changes to pro B cell, not shown here, then becomes pre B cell. This is an important stage of maturation where mu heavy chain is expressed on the surface of the B cell along with the surrogate light chain. And the signaling has to go through that pre-B cell receptor for that B cell to survive longer so that it can undergo further maturation. So important step to remember. All right, we talked about the half-life of antibodies, how it's pretty short for most of the antibodies, EAM, 2 days for E, 3 to 6 days for A, 4 to 5 days for IgM. IgG has a long half-life of 3 to 4 weeks. IgG3 actually has shorter half-life than G1, G2, and G4 subclasses. So important part to remember, clinically it's important because in a newborn baby, if you are trying to understand if the antibodies are coming from mom or these are baby's antibodies, you can look for IgG3. All right, and so what gives IgG its long half-life? So there is something called FcRn or neonatal Fc receptors. The FcRn has a very similar structure to MHC class 1. These receptors are found on endothelial cells, macrophages, several other cell types. They bind to IgG in acidic endosomes, which it means is, say there are antibodies in the circulation, those will be taken up into the by pinocytosis. It basically takes small sips of the fluid where it takes up those immunoglobulins inside the cell, and because of that FcRn, those FcRn will keep the IgG bound to it in these acidic endosomes. In the acidic endosomes, when the IgG is bound to FcRn, it will not be targeted for degradation by lysosomes. So then those immunoglobulin molecules bound to FcRn are recycled to the surface and released into neutral pH of the circulation, right? So the endosomes are acidic and the pH in the circulation is neutral. So what happens is they get bound to the FcRn in the endosomes, and when they recycle those to the surface, they will be released into the circulation. So these immunoglobulins are not degraded by lysosomes, because of which they have longer half-life. The clinical application of this is that the Fc region of the immunoglobulin G can be used to give longer half-life to medications such as TNF receptor immunoglobulin or CTLA4 immunoglobulin. These are fusion proteins that are used for treatment. And just like the monoclonal antibodies, these fusion proteins that are used in clinical practice have a suffix. These will end in sept. So etanercept, abetacept, these are different fusion molecules that are used in clinical practice. So TNF receptor fusion protein, etanercept, or abetacept, which is CTLA4 immunoglobulins, are used for various immune or rheumatological conditions and they are it's convenient because they have a longer half-life. All right, we talked about antibodies. Now we talk about antigens. Antigens are substances that can specifically bind to the antibody or T cell receptor, so any adaptive immune receptor. Immunogens are a type of antigen. So antigens that are capable of stimulating immune response are called immunogens. Hapten is a small molecule. It's an antigen, but it's not an immunogen, right? So antigen can be a hapten or an immunogen. Hapten, when bound to a carrier is a large molecule to which hapten can be conjugated. When hapten is bound to carrier, it can produce immune response. The portion of the antigen or a macromolecule that can bind to the antibody is called determinant or epitope. So part of that antigen that comes in physical contact with the antibody is the determinant or epitope, and those are interchangeable terms. So let's talk about those determinants or epitopes. So the epitopes on an antigen. So let me see if I can show you. Okay, so here is a macromolecule. This is a polypeptide chain that is folded in a certain way. And because of that folding, it gives rise to a structure that can bind to an antigen. So it's a conformational epitope, which means that if it's denatured, that structure or folding is lost, and that conformational epitope will be lost. Compared to a linear epitope, which it doesn't matter if it's denatured or not, that part of the linear epitope is going to stay the same and still be able to bind to the antibodies. And then there are some epitopes called neoantigenic epitopes, which means in its natural form, the epitope is absent. But when it gets denatured or proteolized, there's proteolysis of that protein. There are some parts that are broken down, because of which then there is a new epitope that's created because of that proteolysis. So that's called a neoantigenic epitope. So conformational epitope is because of that folding, and on denaturation, that epitope will be lost. Neoantigen, it's not present, but on proteolysis, there will be an epitope. And then linear epitope, doesn't matter if it's denatured or not, that is going to continue to be present in both the native and the denatured protein. Some of the determinants can be overlapping onto each other, versus the ones that are not are called non-overlapping determinants. There is something called allosteric effects. So binding of an antibody to the antigen, causing a conformational change in the structure of antigen that influences the binding of a second antibody to another site, but by means other than steric interference. So if there are overlapping determinants and one antigen of that overlapping determinant is bound, the other cannot be bound by that another antibody. That's called overlapping antibody determinants, right? Now, when we talk of, that's basically a steric hindrance. That means that if one is bound, the other cannot bind because it's not possible. But something called allosteric effect means that it has nothing to do with binding of with overlap, but because one antibody is bound to an antigen structure, it influences the binding of the second antibody to another site by means other than steric hindrance. All right, for antigen binding, there are two different concepts called affinity and avidity. Affinity is the strength of the binding between a single combining site of an antibody and an epitope of an antigen. So one antigen binding site and the epitope of that antigen bind to each other, and the strength by which it binds to it is called affinity. Avidity, on the other side, is the strength of attachment of all available epitopes to the antibody binding sites, or FABs. This should be antigen binding sites. So the avidity can differ based on how many molecules of antibodies are present. So IgG versus IgM. So IgM would have very high avidity compared to IgG or other monovalent or bivalent antibodies. Compared to affinity, which could be pretty similar, well, depending on how easy it is to separate an antigen and antibody complex into its constituents, it gives the number called dissociation constant. And smaller the dissociation constant, stronger or is the bond, which means there is higher affinity. Lower concentration is needed for complex formation when there is smaller dissociation constant. We have learned this in medical school. When there are polyvalent antigens, it depends on how much antigen and how much antibody is present to determine how big the complexes will be. So if there is too much of antigen, the antibodies are dispersed and can bind to a few of those antigens one at a time, so they don't form a complex. If there is too much of an antibody, they will compete for the antigen, so that a lot of antibody will not be able to bind the antigen, so that again a bigger complex is not formed. So there is something called zone of equivalence, where the antigens can bind to multiple antibodies using those epitopes that are present in equivalent amount, and that gives rise to larger immune complexes that can be formed. Okay, just looking at the time here. Here we are looking at the structure and functional relationships in antibody molecules. Let's look at the original antibody here, and depending on the changes in the antibody structure, we can see how the affinity can change and the effector functions can change. But overall, each of the antibody or each of the B cell will give rise to antibodies of a particular specificity. However, there can be cross-reaction between certain antibodies binding to similar antigens. Overall, antibodies in a human body can bind to various different macromolecules, and overall the diversity that it can bind to is called antigen repertoire. When the antibodies undergo hypermutation in the variable regions of the antibody up here, it can give rise to changes in the affinity called affinity maturation. This will help to improve its binding to the antigen. So it gives rise to increased and improved affinity and called affinity maturation. But the effector function doesn't change. On the other hand, if there is change in the membrane or secreted form, so the tailpiece changes it from the membrane bound, it can get to a secreted form. So there can be change from B cell receptor function to effector function. And then if there is a change in the Fc region of the antibody, it can change the class from being IgM to IgG, A, E, or A or E. This is called isotype switching. And here, the antigen binding or recognition doesn't change, but the effector function changes. All right, that was the last slide. Couple of questions. Which one of the following is a correct description of the basic symmetric core structure of an IgG antibody? Anyone?
Isn't it too heavy, too light?
Two heavy chains, two light chains. Answer is D. An antigen binding site of an IgG antibody molecule is composed of which of the following? D. D is correct. 6 hypervariable loops, 3 on heavy chain and 3 on light chain. All right.